Hi, this is Rishi. Welcome to my channel. Today's topic is Z protein coupled receptors. So before moving to this big topic, let's make a points about the receptors. So what is receptors? So receptors, assume this is one of the cell of your body. So body is made up of millions of cells, right? We have taken one cell. This is obviously we are every cell having a cell membrane. So this is a cell membrane. Okay. On the cell membrane, you will find a number of receptors. Different type of receptors. Different types of receptors. These are the receptors. Okay. Receptors are made up of proteins which is present on the cell membrane, which is responsible for the biological response. So what are the responses, what are the physiological responses you will get with the help of the activation of particular receptor? Okay, got what is receptor? So how you are getting the biological response? Check it. So assume this is a cell membrane. Okay, so I'll take one kind of receptor here. This is a receptor. So I told that whenever the activation of receptors, then only the particular biological response will occur, right? So for activation of any kind of receptors or any type of receptors, we need one signaling molecule or any ligand molecule. Okay, so assume that one of the ligand molecule will come and bind here exactly into the binding site of this receptor. Then this particular receptor get conformational change and get this particular receptor get activated and creates secondary messengers inside. In the cytoplasm region, they create secondary messengers and they undergoes lots of a series of activations. Then one of the molecule, it will cross the nucleus and it will make some alterations in the gene expression get altered. Gene expression get altered, then finally the biological response will occur. Okay, after this particular gene expression get altered, it will create mRNA, right? Messenger RNA. It will come out from the nucleus and it will make some series of actions in the cytoplasm region. They form different particular type of protein. This protein leads to the biological response. Okay, this is the receptor concept. Got? Then we'll move to the specific Z protein coupled receptors. What are the secondary messengers they will create? What are the series of activation that takes place in the particular type of receptors? Okay, then how the biological response will occur, we'll see. Okay, Z protein coupled receptors. In these receptors, we are having two pathways. One is to get the biological response. One is cyclic AMP dependent pathway. Cyclic adenosine monophosphate, and another pathway is IP3 or DAC pathway. Inositol triphosphate or diacyl glycerol phosphates. So you have to know about cyclic AMP and IP3, nor DAG, or all secondary messengers. Okay. You will find in this G protein coupled receptor mechanism, you will find these two pathways. Cyclic AMP dependent path is responsible for one of the biological response. IP3 or DAG path is responsible for to make another biological response. All these two pathways, we are having two major pathways in the GPCR. So GPCR, G protein coupled receptors. So what will happen if a receptor gets activated or created some secondary messengers? It will make some gene alteration, gene expression alteration, then we get the biological response. What is biological response? See, whenever this GPCR get activated, it will make finally a biological response. The biological response is it will regulate the immune system. So one of the major function of GPCR is regulating the immune system. Whenever you need white blood cells or any kind of defending cells, these pathways get activated to create for regulation of immune system. For cell growth, how your cells grow, whenever you are in childhood days, you are getting height and you will get some lots of cells get growth will happen, the production of cells depending upon the Z protein coupled receptor. Whenever these pathways get activated or these pathways is going on, you will finally the biological response will get the cellular growth, the cellular development, everything. So main part is how your taste sensation will happen. You will taste the particular food items, you will sense the taste, right? Because of the Z protein coupled receptors. That's why you have to know about Z protein coupled receptors mechanism. We are having two types of pathway mechanisms: cyclic AMP dependent pathway and IP3 or DAG pathway. Let's see cyclic AMP dependent pathway. So G protein coupled receptors, that means these particular receptors is made up of proteins. So every type of receptor is made up of proteins. Specifically this type of receptors is made up of G protein, guanine protein. Okay, we'll see. So assume that this is the cell membrane. Okay, I'll make you some clear. Cell membrane, this is the extracellular region, this is the intracellular, okay, cytosol region. So this is the cytoplasm, okay. This is the outside of the cell, this is inside of the cell, okay. Z protein coupled receptors, it is also known as 7 transmembrane protein receptors. Why they call 7 transmembrane protein means this type of receptor is made up of 7 transmembrane. So count it: 1, 2, 3, 4, 5, 6, 7. Okay, this is the binding site here outside of the cell. This is the binding site of Z protein coupled receptors. A ligand molecule will give some signal to the particular type of receptors. Okay. So you have to know about where is the Z protein, the guanine protein, the subunits. That means here to the end of this type of receptor inside of the cell, you will find some 3 subunits like this. Okay, this one is I will make it some more clear. This is alpha, this is beta, this is gamma. These 3 subunits combinedly known as Z protein. Okay. So initially, before giving signal to this type of receptors, the receptor structure is like this. Before the receptor conformation changes. Okay. So 7 transmembrane protein. To the end of this type of receptors inside of the cell, we are having alpha, beta, gamma subunits. To the alpha subunit, ZDP is attached, gone in diphosphates. Okay, that's why this combinedly these subunits is known as Z protein. This is a coupled type of receptor. That's why they kept the name Z protein coupled type of receptors. Okay. Let's see. So the tips of these two subunits is attached to the cell membranes. Okay, gamma and alpha. Okay. So whenever so you need some biological responses normally, right? So how these receptors get activated, how these receptors make some conformation changes after binding the ligand molecule. Let's see. So here, so one of the signaling molecule, this come and bind exactly here. Okay, outside of the cell. After this binding, it is giving a first messenger, that is it is giving some signal to these receptors. First message it comes from outside of the cell. So that's why all the ligand molecules or the drug, what are the drug you are taking, those are all first messengers. Okay, ligand molecules. So this is the ligand bind to the particular Z protein coupled receptor. It gets some, the receptors gets activated, these receptors get conformational changes. This GDP get phosphorylated. So one of the phosphate group will come and bind here. It will get activated. This alpha subunit get activated. See. So this is the alpha subunit. So get phosphorylated. One phosphorus group is added, becomes CTP. Okay, guanine triphosphate. These alpha subunits get activated whenever the phosphorylation happens here at this right position. This alpha subunit get dissociated from the other subunits. So what this why we need this dissociation of from these subunits means this part, this alpha subunit, the activated alpha subunit will go and activate other proteins so that it will create the secondary messengers. I told, right, after giving a signal outside of the cell to the particular receptor, it will make some conformational changes. It will create the secondary messengers. So here in the Z protein coupled type of receptors, after phosphorylation to this ZDP, it converts to GTP. It will go and activate another kind of protein which is present inside of the cell. So this is AC, adenyl cyclase. Okay, so here we are having binding site here, exactly come and bind here. So here in this region it will go and activate this adenyl cyclase. It by using the energy ATP, by using the energy, it will create adenine monophosphate, cyclic adenine monophosphate. So this is the one of the secondary messengers, plays a major role in your body, cyclic AMP. Okay. So how these secondary messengers are created? Lots of secondary messengers will create. So we'll take secondary messengers. So let's come from the first. So whenever the signaling molecule or ligand molecule gets bind to the Z protein type of receptors, the alpha subunit, the ZDP, get phosphorylated. Due to this phosphorylation, alpha subunit get dissociated from the remaining subunits. This combined part of alpha and Z protein will go and activate the adenyl cyclase. Whenever this adenyl cyclase is get activated, it receives lots of energy, ATP, to create the cyclic AMP, the secondary messengers. So whenever this part, this alpha subunit, the activated alpha subunit will give the signal to adenyl cyclase, it will create the cyclic AMP. It will, when lots of alpha subunits will go and give the signal to the adenyl cyclase for the formation of secondary messengers like cyclic AMP. Okay, I told that lots of, so whenever this adenyl cyclase create, it will create the cyclic AMP. After that, here dephosphorylation will occur. See here, it will convert to ZTP to ZDP. Dephosphorylation, the hydrolyzation happens here in the ZTP region. Then it will convert to GDP, guanine diphosphate. Then again is alpha subunit and ZDP will go and reattach to the it will take the reposition. It will attach to the beta subunit. So whenever the signaling molecule will come and bind here, it will get conformation, the receptor get conformational changes, phosphorylation will happen, it will convert to GTP, alpha subunit will dissociate and will give the signaling molecule. After giving the signal to the adenyl cyclase, again the hydrolyzation will happen, hydrolysis will happen, dephosphorylation will happen. Again this alpha subunit will go and it will take the reposition of this particular alpha subunit place. Okay, like that, it will give the signal and will go to back to the their position. Again, it will give the phosphorylation. Again, it will give the signal. Again, it will go to the their place. Again, the signaling molecule. Like that. Whenever we need lots of cyclic AMP secondary messengers, at that time, lots of signaling molecule will come and bind to this particular type of receptor for the formation of cyclic AMP secondary messenger. Okay. Then after formation of these secondary messengers, we need to change in the nucleus, in the gene expression get altered, right? This secondary messenger structure, or directly these secondary messengers, so assume this is the nucleus. They can't cross this nuclear membrane. This type of secondary messengers, or the structures of this signaling molecules, can't cross this nucleus. They need another series of activations, right? Secondary messengers will go and activate the protein kinase, which is present, which is flow in the cytoplasm region. Protein kinase. So protein kinase. So they are having like this. The structure of protein, assume the structure of protein kinase like this. So protein kinase is made up of two types of regulatory proteins and two types of catalytic proteins. Okay, catalytes. So for activation of this protein kinase, we need 4 cyclic AMP secondary messengers. So assume this is the secondary messengers we have formed. So the secondary messenger's binding site is present here. See, exactly here. 1, 2, 3, and 4. So whenever these secondary messengers go and bind to their respective binding site, which is present on the protein kinase, specifically in the regulatory subunits, then these catalytic subunits get dissociate. This will get activated. Regulatory protein subunits get activated. Then this particular catalytic subunit get dissociate from this protein kinase structure. This catalase having a capacity to cross the nuclear membrane. This will make gene expression alter. Okay. So this is the gene expression. So catalytic subunits will cross the cell membrane. Okay, I'll make some more clear. Okay, it will cross like this. Then what these subunits will do, will go and bind to the specific proteins which is present in the nucleus. Okay, correct. Cyclic AMP response element binding site. Okay, cyclic AMP response element binding protein. We are having this is a binding site here, exactly here. These catalytes will bind here. It will get phosphorylated. This CREB protein gets phosphorylated. CREB. Due to the catalytic subunits, it will bind to the specific CREB protein. It will get phosphorylated. Then this phosphorylated CREB will make the gene expression alter. So here, this is the CREB. Get phosphorus. It will make some gene expression alter. It means changing the gene expressions. It will make the changes in the DNA regions. It will make the changes in the positions. Okay, amino acids positions, and it will create the new mRNA, messenger RNA. Whenever the gene expression get altered, it will create the messenger RNA. Okay, ribonucleic acid, mRNA. So specifically messenger RNA. Okay. Then, as you know very well, it will move out from the nucleus through nuclear pores. It will come to the cytoplasm region. It will move to the endoplasmic reticulum. It will make the protein synthesis will happen finally. Okay. Then it will go to the Golgi apparatus for the protein package and the dressing up of the protein. Finally, this protein responsible for biological response or physiological response. Specifically, G protein coupled receptors are more present in your body. Is more than 800 G protein type of receptors are present. Okay, I'll tell one example. Whenever your heart get contract and relax will happen, right, in the CVS region, the heart will get contract and relax, contract and relax. For the contraction of your heart muscles, in the heart muscles, we are having G protein coupled receptors. For the contraction of heart, we need cyclic AMP messengers, secondary messengers. Okay. Without cyclic AMP secondary messenger production, your heart will not get contract. So this is the best example. continuously in your body cyclic AMP protection will happen due to the stimulation of one signaling molecule. So I have explained in deep in the molecular level what exactly happen in what will happen in the Z protein coupled receptors. Okay, let's check it out. Whenever the signaling molecule gets contact with the Z protein type of coupled receptors, these receptors get conformational changes. Here we are having in the end of the G protein type of receptors. For here phosphorylation will happen, then it will get dissociated. The alpha subunit will get activated and it dissociates from their respective subunits. Of this activated alpha subunits will go and activate the adenyl cyclase. But adenyl cyclase, whenever the adenyl cyclase get activated, it takes lots of ATP or energy for production of cyclic AMP. Whenever the cyclic AMP secondary messengers are created in the cytoplasm region, it can't move directly into it. It can't cross the nuclear membrane, so that it will go and activate the protein kinase. Here we are having the protein kinase, regulatory and catalytic subunits. 4 cyclic AMP messenger. We need 4 cyclic AMP secondary messengers for dissociation of catalytic subunits. It will only cross the nuclear membrane to activate the CREB protein. This is cyclic AMP response element binding protein. Then it will get phosphorylated. This phosphorylated CREB protein will go and make the gene expression alter. It will change the position for the creating a new mRNA. Whenever the new mRNA is created, it will move out from the nucleus region through nuclear pore. It will go to the endoplasmic reticulum, ribosome. It will create the protein synthesis. Okay. After creation of protein, it will go to the Golgi apparatus for the protein packaging and dressing up of the protein. That exact type of protein will give the biological response. Okay. So here G protein is completely depending on cyclic AMP. So we are having 2 types of pathways in the G protein coupled receptors. One is, you have seen, this is the cyclic AMP dependent pathway. So this is the cyclic AMP dependent pathway. This pathway is completely dependent on cyclic AMP. Without cyclic AMP, you won't get the biological response. So cyclic AMP dependent pathway. And one more pathway is IP3 or DAC pathway. Okay, inositol triphosphate or diacyl glycerol. These are one of the secondary messengers. Okay. Z protein coupled receptors, you are having 2 types of pathways. In GPCR, we can call it as also GPCR, Z protein coupled receptors. In GPCR, we are having 2 types of pathway mechanisms. One is cyclic AMP dependent pathway and another is IP3 DAC pathway. Okay. I'll explain about so this is about the cyclic AMP dependent pathway. I'll explain IP3 or DAC pathway in the next later video. Okay, bye. If you got the concept about the cyclic AMP dependent pathway, please like and share the video. Subscribe my channel for later videos. Okay, bye. Have a nice time.